Hello. Today we're going to look at how prokaryotes control gene expression. We should all understand that a bacteria has a bunch of different chemical reactions that are taking place within it at all times. And it needs to control what reactions are taking place, what enzymes are present, what enzymes are being used because of what's going on in its environment. And it has to be able to respond quickly to those changes in its environment. If it can't respond quickly, that means it has energy that's wasted, and more energy that's wasted means it probably will not survive. Sometimes a bacteria will need to stop a specific chemical reaction. This is typically when they have enough of that product already produced. If they already have enough of the product, they don't want to waste the energy to produce more of that product. And the way to do that is to stop the enzymes from causing more of that product to be synthesized. Or sometimes they need to turn on specific genes to be expressed, so that way they can actually utilize food or a resource quickly for metabolism, growth, reproduction, typical life processes. And the way that they do that is to start the enzymes to be produced for those specific reactions to take place. So a bacteria needs to be able to control what reactions are taking place within it so it can maintain homeostasis, and it needs to control those reactions well, otherwise it's wasteful of energy and wasteful of product. You might remember this, where we had feedback inhibition, where a product would act as an allosteric inhibitor for the biochemical pathway. So in this case, we would have a gene which would obviously be translated for a specific enzyme, and this would then produce a product which could then go into the next enzyme that was being translated for, and then following suit, where each gene is being translated until we end up with our final product. And our final product was an inhibitor for an earlier enzyme. And in this case, it's an inhibitor for the first enzyme that was produced. The problem with this is very simple. It's wasteful. We have made all these enzymes that are not going to be used. And this is not good for the prokaryote because the prokaryote needs to conserve its energy. It doesn't want to waste product to make extra stuff that it's not going to use. So wouldn't it be even better if instead of building all of those enzymes, we were able to block building those enzymes in the first place? What if that product could cause inhibition straight through by stopping the transcription and stopping the translation of each of those genes? This is how the prokaryotes save energy. They don't end up transcribing and translating enzymes that they're not going to be needing at that specific moment. So how does that actually take place? Well, here's how it works. They turn genes on and they turn genes off. For us, we're going to be looking at an example with tryptophan, where the prokaryote turns the specific set of genes off when it has enough tryptophan present. On the other hand, we're also going to be looking on when the prokaryotes turn the genes on. And in this case that we're going to look at, it's going to be dealing with lactose. And when lactose is present, they will turn on the digestive mechanism so that they can digest the lactose. It's based upon a set of genes which are called an operon. And those are genes that are grouped together that all perform or all are used in a specific function. So the operon is a grouping of genes all together on the chromosome that are all going to be used for a specific purpose. All the genes have to be expressed for the bacteria to produce or to perform that specific function. And if any of those genes are turned off, then the bacteria will not be able to perform that specific function. So now we're going to look at it specifically. Here we have our DNA, and it's going to represent our operon. So we're going to have our group of genes that are all put together. And each of these genes will be used to code for a specific enzyme that will be used in a specific metabolic pathway. In this case, it's a pathway to build tryptophan. The promoter, that is where the RNA polymerase is going to bind to. And so it must appear upstream of the genes that are going to be used for the pathway that we are going to be looking at. That single promoter controls transcription for each of the genes for this pathway. They are all transcribed as one unit all together. And there's also going to be something called an operator, which will exist in between the promoter and the genes that are going to be transcribed and then translated. And as we said, RNA polymerase will bind to the promoter. It will then transcribe the entire section of DNA, building the mRNA that is to be translated so that each of the specific enzymes can be made. So when the gene is turned on, the RNA polymerase binds to the promoter and the gene is transcribed as usual, or that mRNA strand is now translated by the ribosome, producing the enzymes which are required for the specific metabolic pathway. Just as before, the substrate is what goes into our first enzyme, and then out of our last, our fifth enzyme, would be our product. So this operon is turned on at this moment. But what happens when you have enough of the product and you don't want to produce it anymore? Then we need to turn these genes off. This is called a repressible operon. Now the product is going to be able to turn off or shut down these specific genes. But it can't do it on its own. It requires a repressor protein, which is going to come about from a gene that is also on the chromosome. And so here we have our gene, which, when it's transcribed and then translated, it builds a repressor protein. The product is able to bind with the repressor protein, which makes it active, where it can now attach to the operator. Once the repressor protein has attached to the operator, RNA polymerase is unable to transcribe those genes. It gets blocked. The other type of operon is called an inducible operon. In an inducible operon, we're going to be looking at specifically a lactose operon, where it's comprised of 3 genes that are all going to work together to make 3 proteins. The lac genes are typically turned off by a repressor protein and are only turned on in the presence of lactose. So this is the exact opposite of the example we just gave with tryptophan, where tryptophan turned off, or the presence of tryptophan turned off that operon. In this case, the presence of lactose is going to turn on this operon. Here are the different parts to it. We have the genes LAC-Z, LAC-Y, LAC-A. They are going to code for the different enzymes. You have the promoter, which is LAC-P, the operator, which is LAC-O, and our regulatory gene, LAC-I. Each part performing a specific function to make sure that those genes are not expressed when lactose is not present. So again, we have our strand of DNA. When lactose is not present, then LAC-I will continuously be transcribed and translated, which will produce a repressor protein which binds to LAC-O. The repressor protein is active without the presence of lactose. It blocks the RNA polymerase, and thus the genes are turned off. But in the presence of lactose, lactose is able to bind to that repressor protein. And when it does, it inactivates it, making it so that the repressor protein cannot bind to the LAC-O. At this moment, nothing is blocking the RNA polymerase from going through and transcribing those genes. This turns them on, and each of those enzymes are then able to be produced through translation. Let's look at this again. Remember, we have repressible operons. They are always on. They can only be turned off in the presence of a specific inducer molecule. Inducible operons are typically off unless they are in the presence of the inducer molecule. In general, what we can look at is that repressible operons are typically for anabolic pathways. In other words, pathways where we are synthesizing our end product, trying to build up our end product. So once the prokaryote has synthesized enough of the end product, the operon must be shut down. And those are the repressible operons. The inducible operons, they are catabolic pathways where the operon is going to be used to break down a molecule for digestion. Those are only turned on when the molecule that is to be digested or broken down is present. This is all done, or this is all used to make sure that the cells avoid making excess enzymes, wasting energy, building molecules that they do not require. That's it for this time. Be awesome, stay awesome.